Hey there, guys. First set of video lecture notes. I'm just wanting you to make sure that you've got your digital notebook paper ready to start taking some notes. So we're going to get started here with the scientific method, okay? So the first thing that you're going to get when it comes to science and the process of looking at the world around you is this process of the scientific method. You probably heard about it in science in junior high, so this should be a review. So just make sure that you are taking good notes and really paying attention to what we have to say about it. So scientific method, basically anytime we want to know something about the world around us, we use the scientific method to investigate it. And there are several steps to the scientific method. And this little flow chart kind of gives you an idea of the steps of the scientific method. We first observe something or we have a question. Then typically in between observe and form hypothesis, we do a little bit of research. We take a look at what we know about that thing already. Then we form our hypothesis, which is basically our prediction of the answer to that question that we're asked before. Then we go into testing our hypothesis, so experimentation. How are we going to go about trying to prove our prediction is correct or incorrect? Then we get results, we analyze the data, and then we evaluate the results and we draw conclusions. So we're going to go through this kind of step by step. So drawing this chart, flow chart, is kind of a good idea, especially if you're a visual learner. It can really help you to put those steps in a flowchart way. If you're a linear person, you could totally put those in bullet points or numbers going in order. And then now we're going to go into each step on its own. So the first step is observations. So scientists make careful observations prior to doing research, and we use all 5 senses to make our observations: sight, sound, smell, taste, and touch. We see something out in the natural world that we are curious about, and we make some observations about that thing, or we have questions about that thing, and we're going to take those observations and then we're going to do a little bit of research after that. So in between this, I need to go back. In between this step 1 of observations and step 2 of forming a hypothesis, you can just insert a little arrow there or something that says that you will do some research. So going from our question, maybe, I don't know, why is the sky blue? At one point, that was an observation or a question. The sky is blue. Why is that? Then the person who thought about that, or persons, had to then go out and do some research into, well, do we know anything about that yet? Do we have any information about that? So always between observations, step 1, and forming your hypothesis, step 2, you always have some research. So you can input that right there. Step 2 is going to be forming your hypothesis. So when we use observations to ask questions about what we are investigating, or when scientists do that, when they use observations to ask questions about what they are investigating, they have to ask questions that are testable. So it can't just be some random, okay? It has to be a question that you could then take and test. So for example, you got a couple of questions here. Does increasing the amount of light a plant receives increase its growth rate? Versus, are plants grown in artificial light look nicer than those grown in sunlight? So we've got like 2 different styles of questions. The first question, does increasing the amount of light a plant receives increase its growth rate? We're talking math, right? We're talking how much does it grow in a certain amount of time? Okay, so we're talking something measurable with like a ruler. The second question asks, are plants grown in or it should say, do plants grow in artificial light look nicer than those grown in sunlight? That's a question of aesthetics, okay? When we observe the plant, does it look nicer? Does it look healthier? So those are 2 different ways that you could test. You could test based off of the math, the growth of the plant, and that question, or you could ask a question based on the look, the perceived health of the plant. So 2 different styles of questions, and both are valid for scientific experimentation. So from those questions, scientists can form predictions about the answer. So you ask a question, for example, let's say, does increasing the amount of light a plant receives increase its growth rate? So if I give a plant more light, will it grow faster? We can form predictions. So we can form an idea of what we think is going to happen, and that is a hypothesis. Anywhere you see that is bolded or underlined or anything like that, you're definitely going to write down, and you should already have in your flashcards already. So that's just another vocab term. So your hypothesis is a proposed answer for a scientific question. So it's your prediction. It's what you believe is going to happen based on your observation. And in this case, if we're talking about growth rate, we can make a hypothesis about that. So personally, if it's me and I'm thinking, does increasing the amount of light a plant receives increase its growth rate? My hypothesis could be something like a plant that receives more light than another plant will grow at a faster rate. That would be my prediction. Okay, so that would be a hypothesis. So then step 3, you're going to test the hypothesis, and that's done by experiment. Okay, so here it says testing hypothesis. You can put /experiment. Okay, so step 3, you're basically going to test that hypothesis. Scientists collect data that they use to support or reject a hypothesis through experimentation. So once I've made an observation and I've come up with a prediction, and in between those two, I've done a little bit of research, I'm now going to design an experiment. I'm going to test or figure out a way to test what my question is and to see if my prediction is proved true or not. Okay, so scientists collect data that they use to support or reject a hypothesis through an experiment. Okay, so if I want to go back to the plant, if getting more sunlight affects the growth rate or not, I can now go get a couple of plants, put one in no sunlight, one in regular sunlight, and one in more sunlight, and then I can test and see what does sunlight do to the growth rate of that plant. That would be my experiment. So a couple of different parts of an experiment, and you're going to want to write these down. And again, these should be in your flashcards, so this should be the second time you're seeing these, okay? First of all, we have variables. So in an experiment, you have variables. You have something called the independent variable, which is the thing that is manipulated or changed by a scientist. Okay, so independent variable is what is changed. It's what you are doing to the experiment. The dependent variable is what is observed and measured during an experiment. It's the experimental data. So in this case, I always tell my students, dependent is data, okay? Dependent is data. It's what you're going to measure. In the case of our plant and whether it grows faster or not in more light, the dependent variable would be the data that you're getting out of it, the height of the plant, okay? So dependent is the data that you're measuring after or during the experiment, okay? Dependent data. The independent variable, again, is what you are changing. In this case, it would be the amount of light that that plant is getting, okay? So independent variable, what you are changing, and dependent variable, the data that you are collecting, okay? And then you always have a control group, okay, within your experiment. You've got conditions that do not change. So in the case of our plants, okay, you would have a control group where the conditions don't change, and that would be what you are comparing that to. So a plant in regular amount of sunlight would be your control group in that case, or you could use that as your control group. So a regular plant, regular light, what does it grow like? And then compare the plant that is put into more sunlight, and then you could use that. Okay, so, oops, we need to go back. Another thing that you need to add in here, and I'm sorry that we didn't put in, is that there is the experimental group. So the control group is the group where nothing changes, and the experimental group is the group that has the independent variable, okay? So again, your experimental group, okay, is the group that has the independent variable. It's the group that gets the change. And then your control group or your control is the group that gets no change, okay? All right, so now we have a couple of questions. All right, so variables and controls. So we're going to take a look at an example. And we're just going to do one because we're going to go back through and do this scientific method activity together. And so we're just going to take a look at this first one. And it's from The Simpsons, if you watch that. So Mr. Smithers thinks that a special juice will increase the productivity of workers. He creates 2 groups of 50 workers each and assigns each group the same task. In this case, they're supposed to staple a set of papers. Group A is given special juice to drink while they work, and Group B is not given the special juice. So Mr. Smithers gives a group of people, Group A, a special juice that he thinks is going to increase their productivity, basically make them able to work better, work faster. Okay, and then that's given to group A. And then group B does not get anything. So in this case, what is the independent variable? So you're going to think about this, and you're going to write down the question, what is the independent variable? You're going to think, what is the independent variable? And again, remember, independent variable is the variable that is changed. Okay, so I'll give you like 2 seconds. What is the independent variable in this case? What is changed? 3, 2, 1. In this case, the independent variable is the special juice. So the thing that is being done in the experiment, being given, is the juice. All right, and then so then what would be the dependent variable? What's the data that is going to be measured from this experiment? What is Mr. Smithers going to measure to see if his hypothesis was correct? And in this case, it's the productivity of the workers, okay? So he's measuring the productivity, how fast or how many sets of stapled papers they can do. Did group A do more with the special juice, or did group B do the same as A? How did that work? And then what is the control? So what is the group that is the normal conditions, that doesn't change at all? In this case, it would be group B, right? So group B is the group that doesn't get anything done to them. They're the control group. They are the normal conditions for that environment. They are the normal workers with no adjustments. Group A, okay, the group with the special juice, is your experimental group. They get the independent variable, all right? Okay, so next, after that, after we go through doing our, I'm going to go back really quickly, after we go through step 3 of testing our hypothesis and through an experiment and looking at all of our variables and controls, okay, going back through Mr. Smithers, we are going to then analyze the data. Okay, so you're going to take a look at your data and draw some conclusions about what you did. Right? So there's 2 types of data, or data, whichever you want to say. There's quantitative with an N right here, okay? And then there's qualitative with an L, and I'm going to give you a really simple way to remember that. Quantitative with an N always means numbers. It's data that you're collecting from an experiment that you can measure with a ruler. You can measure with some sort of volume measurement, so you're talking meters, kilometers, miles, inches. However, in science, we always use the metric system. So we're talking meters, kilometers, centimeters, millimeters, something like that, or liters, grams, anything that you can measure with numbers, okay? Quantitative with an N means data that is numbers. So again, first type of data, quantitative data with an N, is numbers with an N. So any kind of data that has a number. When you were looking at the other type of data, qualitative, that's descriptive words. So you're talking more of like describing what the particular thing looks like, smells like, feels like, tastes like, smells like. Things like shiny, large, small. What you can view or sense with your senses, that's qualitative data that you'd be collecting. So it looks healthier, it's shiny, it is smaller than it was before or larger than it was before. So again, you need to be able to distinguish between the 2 types of data. Quantitative with an N is numbers, meters, liters, grams, and qualitative, the one with the L, I just call it the other one, it's easier for people to remember that way, is something that describes the way something looks, sounds, smells, tastes, all that kind of stuff, okay? When we're evaluating our results, we basically look at our results, and then we also look at the data and conclusions presented by other scientists. So it just helps us to get peer material from other people in order to validate our results. And then if you go through a whole process and you do the whole thing, eventually you can get to where you propose a theory, which in this case is a proposed explanation for a wide range of observations and experimental results that is supported by a wide range of evidence. In order for something to be a theory, there has to be a ton of experimentation that has proven their hypotheses correct and is now something that we take not as law, but as theory, which means we're pretty much 100% sure, maybe not 100%, but mostly sure that it is true. Once you finally get through getting your results, then you're going to start drawing those conclusions, okay? So when you get to your conclusions, you are basically at the kind of towards the end point of the entire scientific method process. So basically this is used to determine if your results support or disprove your hypothesis. So if your hypothesis was proved correct or proved incorrect. So you get through all your experimentation, you get all of your data, and you take a look and you say, okay, based on this data, is that supporting my original hypothesis? Does the data support my hypothesis, support my prediction? Or is it actually the opposite? And does it disprove my prediction? Okay, so in your conclusion, which when we start going through experimentation, you will have some opportunity to be able to kind of write through these. You can restate your hypothesis, so you can say what you originally thought and then conclude what the data told you. Okay, so you are writing out, this is what happened. These are the steps that I took. Here is what I found, and this is what that means in regards to that data and that experiment. Once you get to your conclusion that you've proved true or disproved false your hypothesis, you can take that information for further research or application. So if your hypothesis was proved correct, you can then go, okay, now what can I do with that? Is there something I can do with it scientifically or out in the world? And you can use that for application. If you find that your hypothesis is disproved, you are incorrect, you can use that as a jumping off point to now go do some research and maybe ask another question and see if you can figure out how to make what your original hypothesis was into something new. And that doesn't mean that your hypothesis was a failure or your experimentation was a failure. There's no failed experiments. There's only disproved hypotheses, which is not necessarily at all a bad thing. It just means that now you have something that you know for sure doesn't work, and you can start and go in another direction. Okay? All right, so after an hour, this is a practice conclusion. After an hour, Mr. Smithers counts how many stacks of papers each group has made. So remember, this goes back to group A and group B. One had the special juice, one didn't. Remember, group A was the experimental group, and they had the special juice, so they had the independent variable. And group B was a control group, no juice, okay? And group A made 1,587, and group B made 2,113 stacks. So right now I want you to think, what could you conclude from the experiment? Looking at the productivity numbers, which is our data we're getting out of the experiment, and it's quantitative data because it's numbers and n because we're counting stacks, okay? What happened in this experiment? Was Mr. Smithers' hypothesis proved correct? He thought giving the special juice would make them more productive, okay? And group A had the special juice and group B did not. So what can you conclude? Okay, and that will bring us to the end of our PowerPoint at this point. We are in a place where we can get to our ending. I'm going to go back and just take us back to the flow chart. If at any point you have other questions, there is another video I'm going to post on your Google Classroom. It's a really short 4-minute teacher's pet video. I highly recommend that you go to that at the end of this lesson and add anything from that video onto the end of these notes. And it gives you an example of experimentation using your cell phone. So that might be a little bit more to your liking, and you can use it to flesh out the rest of your notes. So I hope that you guys had an easy time getting through the video, and we will see you in class next time. See ya.